Acute kidney injury, AKI, is characterized by a rapid, often reversible loss of renal function. Current AKI mortality remains around 50%. There is no effective treatment for AKI that accelerates recovery. The only treatment is symptomatic and consists of replacement of renal function by dialysis in severe cases. Chronic kidney disease, CKD, is among the fastest growing global causes of death. It is projected to become the fifth global cause of death by 2040 and the second cause of death in long-lived countries before the end of the century. Therapeutic options to prevent CKD progression are limited, contributing to the high burden of disease. Furthermore, chronic kidney disease is the condition that most increases the risk of lethal COVID-19. AKI episodes favor CKD progression, and CKD is a risk factor of AKI. Thus, it is imperative to develop new therapeutic options for AKI and CKD. Mitochondrial biogenesis is a complex process that involves the replication of mitochondrial DNA, the synthesis of the mitochondrial membranes, and the mitochondrial encoded proteins, and also the synthesis and import of nuclear encoded mitochondrial proteins, requiring a coordinated regulation of the two distinct genomes. In this context, PGC1-alpha is the master regulator of mitochondrial biogenesis that coordinates the transcriptional machinery, leading to increased mitochondrial mass. Through its interaction with additional nuclear factors, PGC1-alpha regulates multiple pathways involved in cellular energy metabolism, implicating processes like mitochondrial fatty acid oxidation, lipogenesis, thermogenesis, and glucose metabolism, which are critical for tissues with high metabolic demands, such as heart, skeletal muscle, brain, and kidney. Renal tubular cells consume huge amounts of energy to reabsorb the bulk of the glomerular ultrafiltrate. So mitochondrial dysfunction is a key factor in the pathogenesis of AKI, causing tubular cell dysfunction and death. We now review the contribution to kidney disease of the altered expression and activity of PGC1-alpha.
PGC1-alpha has positive and negative regulators. Inflammatory mediators such as TNF-alpha or TRIC reduce PGC1-alpha expression through activation of nuclear factor kappa B. In a sepsis model, LPS mediates the activation of MAP kinases and promotes PGC1-alpha downregulation. Furthermore, prophylactic factors may also reduce PGC1-alpha expression. In contrast, sirtuins, AMPK, stone proteinylation, and other factors promote PGC1-alpha upregulation. There is functional in vivo evidence of the role of insufficient PGC1-alpha activation or even PGC1-alpha downregulation in the pathogenesis of diverse forms of AKI, as well as of the therapeutic impact of preserving PGC1-alpha expression in experimental AKI.
AKI induced by different insults, as ischemia, sepsis, and nephrotoxics, is characterized by a decrease in PGC1-alpha gene expression and activity, leading to downregulation of PGC1-alpha target genes and inflammation. Additionally, genetic PGC1-alpha deficiency is associated with spontaneous evidence of subclinical injury, as well as with increased sensitivity to AKI. In contrast, PGC1-alpha overexpression in tubular cells is associated with decreased severity of experimental AKI. The role of PGC1-alpha in CKD and potential therapeutic approaches to preserve PGC1-alpha activity has been most extensively studied in diabetic nephropathy, where PGC1-alpha levels are decreased. PGC1-alpha overexpression prevented mitochondrial dysfunction and cell death in cultured human proximal tubular cells in a diabetic milieu. Sirtuin-1 agonists increased PGC1-alpha levels and protect against high glucose-mediated mitochondrial injury in cultured podocytes. However, forced PGC1-alpha overexpression in podocytes caused collapsing glomerulopathy, suggesting that a tight regulation in at least some cell types is required. Moreover, fibrosis is a key process in CKD progression, and PGC1-alpha was downregulated in different murine models of renal fibrosis, such as not transgenic mice. In conclusion, low PGC1-alpha activity appears to be a common feature of AKI and CKD, and it seems promoted by inflammatory and fibrotic processes. The functional impact of PGC1-alpha has been conclusively demonstrated in preclinical kidney injury, as PGC1-alpha deficiency in either whole kidney or specific kidney cell types was deleterious. Contrary, genetic PGC1-alpha overexpression was generally protective. However, this therapeutic approach is unlikely to reach the clinic. Thus, of even more interest is the characterization of drivers of PGC1 alpha downregulation or activation that can be targeted therapeutically. These nephroprotective approaches that increase PGC1 alpha activity may pave the way for nephroprotective strategies potentially effective in both AKI and CKD.